My name's Deborah Baines, and I'm a Professor of Molecular Physiology at St George's University of London. About 10% of people with cystic fibrosis are currently not eligible for modulator therapy because they harbour variations in their CFTR genes which don't respond to the modulator therapies. So genetic therapeutics, gene editing and gene therapy offer potential therapies for these people, but they're currently quite inefficient. So we hypothesised that if we could replace the CFTR gene not only with a corrected version, but also one that was more efficiently made in the cells or was actually more active, then this would help make the CFTR more active in the lung.
Specifically in our study, we looked at optimizing gene therapy strategies by using different variants of CFTR. We used codon-optimized forms of CFTR that can produce more protein, and we also looked at gain-of-function mutants of CFTR that can increase the amount of chloride that is able to pass through the channel. Interestingly, we found that the codon-optimized CFTRs did indeed produce a lot more protein in cell lines. However, this did not translate to primary cells, where actually function was decreased compared to wild type. This may be due to mislocalization of the protein or overproduction of the protein, leading to inhibition of vectorial transport. So our findings suggest that codon-optimized forms of CFTR may not be suitable for gene therapy strategies. However, gain-of-function mutants, like the K978C CFTR that we use in this study, may be beneficial. The importance of this study is it gives hope to gene therapy practices, which are currently very inefficient in vivo. So using a form of CFTR that is able to restore function more effectively than even the wild-type version could enable better treatments with gene therapy in the future.